Hi, everyone. Thanks for taking some time out of your day to join us for our first BRIM webinar. This is going to serve as the launch for our mining microbiome theme, and we're really excited to show what this theme has in store. So our webinar titled Biotechnology for Exploration, Extraction, and Remediation will feature 5 BRIM researchers. So we'll start with Dr. Stephen Hallam, who will talk about metagenomics tools, and then Dr. Rachel Simister, microorganisms as sensors for concealed mineral deposits, Rob Greene, biochemistry for mineral processing reagents, and then Dr. Vikram Yadav, extraction of low-grade copper and remediation of acid mine drainage through sustainable bioprocesses. And finally, we'll end with Dr. Sue Baldwin talking about microbial communities contribution to mine remediation. So this will be recorded and it will be available on our website after the fact. If you have any friends or colleagues who are unable to make it, that will be available. And if you have any questions, we have the Q&A that will just go to the panelist and myself, and I'll read all those questions out at the end. And there's also the chat feature, which I'll also keep an eye on, but then you can communicate with each other as well via that. I think that's the lay of the land. It should be at about 60 minutes of presentations, 30 minutes of questions, and I will pass it on to the new BRIM director, John Steen, to say a few things.
Thanks, Dana. Dana is BRIM's administrative manager and does a fantastic job in coordinating BRIM's activities and in events like this. So thank you, Dana. Welcome, everybody. This is BRIM's launch of the mining microbiome research theme. I'll say a bit more about BRIM and BRIM's objectives and how we got here in a minute, but just very quickly, being a mining related research centre, this is my safety share. You can see my safety cougar. I think one of the things we're figuring out about COVID and responding effectively is the importance of leadership and communication, constant communication about safety, and reinforcing the message consistently and in different ways. So if you go to the BC parks at the moment, you'll see all sorts of animals that are 2 meters long. So someone said to me, if you see a 2 meter cougar, probably the COVID is the worst of your safety problems. So they might have a point there. Okay, so a little bit about BRIM. BRIM is a research institute within UBC. It stands for the Bradshaw Research Initiative in Mining and Minerals. The Bradshaw name is Dr. Peter Bradshaw, Canadian mining identity. If you want to learn a little bit more about Peter, please Google Peter Bradshaw Canadian Mining Hall of Fame, and you'll see an entry there about Peter's work over his career. UBC has a very long history with mining-related education and research. We have many different activities across a very large campus in mining. Just to pick a few examples, the Mineral Deposit Research Unit, the Exploration group, which does industry collaboration. They celebrate their 30th anniversary this year, which is for an industry-funded centre is quite remarkable. And if you've been following the media, and particularly the Financial Times, you would have probably read about a very high-profile mining company called Yeti, and they use they're talking about a new technology for processing copper, which is much less energy intensive, much less polluting, much less, it's really a big breakthrough in green production. And that technology comes out of UBC too. That patent is held by UBC researchers out of the materials engineering group, in particular the hydrometallurgical group. So if you want to Google hydromets, they're part of the UBC mining research network. What we haven't done so well, I guess, over the years is bring these disparate groups of expertise together to really coordinate activities around the big challenges in mining. And Peter Bradshaw saw this as well, and he said, look, I'd really like to assemble the expertise and bring it together to work on collaborative projects. And this is what BRIM does. So you'll see that with the theme that we've brought together to launch today, the Microbiome Biotechnology, it represents experts from across UBC in different departments and different faculties. But also, very importantly, if you look at it, we're really covering from, the exploration, the beginning of the mining cycle, right through to the closure of the mine and the remediation of the site. And that's another objective of BRIM, to really cover the whole of the mine life cycle. So what we'd like to get out today, what our objectives are really, is to showcase this expertise. And the other objective is really to, show you out there in the industry and government research centre, government, what we're doing, and really seek partnerships. The way that BRIM mobilises UBC research and gets it out there to have impact is through collaborative partnerships. So that your part in this is essential. And if anything tweaks your interest, or if there's a conversation that you'd like to have, we're completely open, all ears. Contact us, email, LinkedIn, whatever suits you best, and we'll respond very quickly. We're very eager to have conversations. So without further ado, I shall hand it over to, who we on first, Professor Hallam or Rachel Simister? Who we got?
On the Stephen Hallam.
Yeah, okay, Professor Stephen Hallam on genomics.
Okay, thanks for the opportunity to talk a little bit about how we can harness the power of metagenomics to build the mine of tomorrow. And I want to reference that term because it actually comes from one of my colleagues who'll be presenting today, Scott Dunbar. And here's a quote from Scott. "Mining's flight to scale is an astonishing human achievement. Some of the world's largest mines have 500,000 tons of rock every day and can be seen from space. But scale has not solved the fundamental problem in mining. As ore bodies become more difficult to find and to mine, productivity declines." And so what we're really interested in conceiving of is a situation where we can harness the power of biotechnology to, in some ways, transform current mining paradigms into more of a bio refining or a biofactory context. Mineralizing rock masses and scrap metal and mine tailings, sort of waste to value processes, so that we can actually recover metals, produce materials and products that have economic value. And what we're going to do today is explore how microbial communities, collectively termed the microcosmos, can be harnessed to realize some of these ideas. We're going to go through the looking glass of metagenomics, specifically how we study microbial communities using high throughput sequencing methods and computational pipelines to interpret that information in a mining context. And then we'll talk a little bit about the concept of a biomining knowledge base that could be used by both academics and practitioners alike to develop a community of practice around microbial biotechnologies in mining. So we live in a world that is dominated by microorganisms. Prokaryotic microorganisms, in particular bacteria and archaea, account for approximately 10 to the 30 cells. This is a very large number. It's more than the number of stars in the known universe. It's more than the collective synapses of all of humanity combined. And it's that power of large numbers that really gives microorganisms their impact on planetary scale processes. This is a wiring diagram of planet Earth metabolism, if you will, the way that reactions connect at planetary scales. And microorganisms mediate all of these processes. So they are the engines that drive Earth's biogeochemical processes and have been transforming the Earth over billions of years of cellular evolution. And they do that through networked metabolism. So this is a network. It could be a social network, a banking network. In this case, it's a microbial network where the links between nodes, which represent microorganisms, the links represent connections. They represent some kind of metabolic association or co-occurrence pattern. And what we really want to understand is how to harness this connectivity pattern to build biotechnologies that work under specific conditions or for specific outcomes. But this is very difficult because we're essentially dealing with uncultured communities of microorganisms, and we can really only access them through their genomic information. And so in effect, we have to convert the network cellular structure into genomic sequence information. We have to learn how to read that matrix of ones and zeros to build those biotechnologies. And so we do that through the lens of biological information. We can look at DNA, which is the hereditary information that records the evolutionary history of the cell. We can look at that through RNA, which is a messenger, which communicates the message from the DNA to the ribosome, which builds proteins, and proteins down here do the work. So we can look at all three levels. We talk about that as multi-omics. When we sequence the microbial community as a total, plurality, we refer that as metagenomics. If we look at the RNA, we refer that to metatranscriptomics. If we look at the protein at that totality level, we call that a metaproteome. So one of the things we can do with the information is build molecular barcodes that help us differentiate between one community of microorganisms and another. And you'll hear from Rachel. Yeah.
Just quickly, we can't actually see your slides. Are you sharing?
Oh, I am. Oh, you know what? I'm so sorry. That I thought that was automatic.
No.
All right, well, let me get in here, and I think I'm sharing now.
We can see now. Yep.
Okay, so I'm just going to pick up right here at the matrix. This is what happens when I can't see the screen fully. So I was talking about biological information, and I was talking about how microbial communities are, for the most part, the ones that we're interested in are not accessible through cultivation. So if we really want to harness the power of microbial communities to build new biotechnologies that will help build that mind of tomorrow, we're going to do that through the lens of genomics. And that involves sequencing the hereditary information of those microbial communities as a totality, as a plurality, as a community. We can also access other forms of biological information. That could be the RNA, which is the expressed information from the DNA that's used to encode proteins. The proteins are then produced at the ribosome, and those are the entities that do work inside the cells. They're the catalysts that we also want to harness. And so we can use that biological information as molecular barcodes, and you'll hear from Rachel how she's using molecular barcoding in an exploration context to look for mineral deposits. You can also use them as biomarkers or diagnostic tools for monitoring the performance of reactors or processes that you're interested in if you're remediating tailings or something like that. You can also use the biological information to reconstruct the metabolic potential of the microbial community. And this is where it gets really interesting from an engineering design perspective. So we can look at these genetic pieces of information and how they fit together to build machines, the catalysts that actually do all of the interesting work. And we can both use those directly from the communities that occur in mining sites. We can also use synthetic biology approaches to build new machines that do specifically what we want. But there are some very big challenges to navigating that matrix of information that we are able to generate now relatively easily at lower cost. And that is that we have to reduce the time to completion of research because the data sets are very large and they're very complicated. That often require many people to work through this information to extract meaning. And so we want to be able to reduce staff and expertise required to read that matrix. And of course, there's end-to-end storage, documentation, and reproducibility issues. So there needs to be a whole platform, in effect, that allows us to convert the biological information into actionable insights. And so that's really what I wanted to touch on today. One of the approaches that my group has been developing over the last almost decade is bioinformatics pipelines that help us read that biological information in the matrix. Here's a pipeline called MetaPathways that takes the sequence information and through a series of stage processing steps renders a database and a metabolic model that can be used to look for interesting catalysts within a microbial community. We call these models environmental pathway genome databases. It's publicly available software. You can find it at GitHub. And it's meant to empower people to interpret and translate the knowledge that is within their sequence information. It has an integrated user interface, and it's also compliant on high-performance computing resources. And so an environmental pathway genome database, this model of metabolism that represents the microbial community, is roughly schematized here. You have the microbial community, and you have the sequence information that's extracted from that community. It's organized in a hierarchy, which is the underlying database structure, and then it can be rendered in different formats, tables or visualizations. So through the graphical user interface, you might see something like this, which is a table where you have columns that represent different samples, and you have accounting. You have the total number of sequences, you have the types of pathways that are present, and you have processing steps in the background. And so the idea is to create a uniform framework for processing information that's also easy to access by the user. You can also visualize that data looking at the community as a metabolic network or a community cellular overview. You can zoom into individual reactions all the way down to the genes that encode them. And so in this way, you're able to look at the community as a cell instead of, all that complexity overwhelming you. I wanted to just briefly show you how we applied that with some data from Sue Baldwin, who also will be presenting today. As you know, there was an incident at the Mount Poly site some time ago, which resulted in spilling of tailings into the environment. And Sue's group has worked over the years to try and find ways to help remediate that. Here's a picture of that dike that broke. And so she provided samples from these biochemical reactors down here that were involved in selenium and sulfate removal, generated DNA sequence information from these different reactors. And we compared the results from those reactors to another type of bioreactor involved in enhanced biological phosphorus removal. So more on the wastewater treatment side of things. And we took all of the sequence information and we created essentially a barcode for each sample. And then we use statistical methods to visualize the relationships between those barcodes. And what you can see here is that there's a nice clean separation between the bioreactors doing the phosphorus removal and an intriguing separation between the two biochemical reactors involved in sulfate and selenium reduction. We can compare the totality of pathways by doing a type of set difference analysis and just say what types of functions are shared between those different reactors and what types of functions are unique. And that might be very important in engineering or improving a process. We might be focusing on those unique pathways that differentiate them. So there was a core of almost 1,000 different pathways that were shared. And then you can see that there were some specific pathways for the two biochemical reactors, about 58 for one and 20 for the other. And then we can zoom in and we can start to look at the differences between those pathways. So they're clustered at the top, and you can see again that those BCRs are differentiated down at the bottom here, these 4 samples, from those involved in enhanced biological phosphorus removal. And then we can specifically ask what types of pathways are unique, those ones at the edges of the Venn diagram. And then we can go in and we can see that there are pathways for phenol degradation, or thiosulfate or sulfite oxidation that are specific to those BCRs that may be relevant to the process that is being engineered. And so we can also take that a step further and apply something called biomining, which is a way of functionally validating pathways that we might identify in silico using the computer by essentially developing screening tools that go after the proteins that are being expressed. And the way we do that is we'll extract the DNA, and instead of sequencing it, we'll build what's called a library of those DNAs. We'll put them into E. coli that we can grow in the lab. And then we'll actually probe for the functions of interest, whether it's sulfur reduction or whether it's selenium detoxification. We can build a screen in the laboratory to do that. We can identify the biocatalysts, and we can ultimately use them in synthetic biology applications to engineer new strains. So the last thing I wanted to bring up was that all of this needs to be done within a knowledge-based framework. And so we would like, through BRIM, to be building an architecture, a cyber infrastructure, if you will, that would collate a lot of the mining-related data sets into a common framework that could be used by both academic and industrial users to improve their processes. So in conclusion, I told you about the microcosmos. There are 10 to the 30 bacteria and archaea on the planet. They're organized in metabolic networks. We can develop new search functions using computers and specialized algorithms to tell us about the genetic composition and metabolic potential. And we can use various functional screening methods to actually build new types of microorganisms that ultimately allow us to build the mine of tomorrow. And with that, I will just say that there are some very exciting things that we can do going forward with the development of tools. And you're basically going to hear about this looking forward during the rest of this webinar. So I'll end on that note. Thank you very much for your attention.
Thank you very much, Steve. Really exciting. I did biochemistry back in the 1980s, and I'm still amazed at how far it's gone. It's a really advanced science now that has lots of industrial applications, so that's very exciting. We had one question, and the question was from someone in Kenya, I believe, and she said, do you know of applications or to treat mercury in mines?
Yeah, so that's actually a very interesting question. I've collaborated with several people who are interested in methyl mercury and its fate in the environment, and we can definitely identify genes that are implicated in that process in different types of environmental samples. And so the short answer is these tools are directly applicable to that problem. Of course, it requires not only analyzing the data, but ultimately validating it. So I would just encourage people to think about that. It's one thing to go looking for the information. It's another thing to actually take that information and action, realize through actions some function. And that's the part that I find most exciting. And I think you'll hear from Scott on one of the ways that they're doing that to actually pull minerals out from complex ores.
Excellent, thank you very much. And we might move on to our next speaker, which I believe is Rachel.
Yep. Let me just
make sure we get the screen share.
Can you guys all see that? It's up.
Yeah, that's great. Over to you.
Cool. Okay, so yep, I'm Rachel Simister. I'm a research associate in the departments of Microbiology and Immunology and Earth, Ocean, Atmospheric Sciences at UBC. And today I'm going to talk about how you can use DNA from microorganisms as sensors for concealed mineral deposits. And so, yeah, Stephen just gave us a great intro in all the wonderful things that you can do with DNA from microorganisms. And yeah, I wanted to kind of reiterate that the kind of thought behind using microbes for mineral exploration came from the fact that, yeah, microorganisms are ubiquitous in almost every environment on Earth. They're highly sensitive and responsive to chemical and physical changes in their environment. And so together, they're largely responsible for catalyzing biogeochemical cycling on Earth. And so I find a really great way to sort of visualize this is to look at phytoplankton, microorganisms in the ocean, responding to trace amounts of iron. So these are like trace concentrations of iron that change, and you can visualize these microbes from satellite imaging from space. And so if we take the same principle through to soil, in a gram of soil, it's estimated that there can be as many as 10 million living organisms, and these comprise tens of thousands of bacterial species, each sensing and interacting with their surrounding soil environment. And so historically, microbiological techniques have tried to be used in mineral exploration, but they'd found limited application. And that was because some of the methods Stephen was talking about, that we didn't really have the technology until very recently, and sequencing used to be quite expensive. I've just got a couple of slides to illustrate that. So this is what the cost of the human genome, how it's changed over time. So if you go back to the early 2000s, you're talking of tens of millions of dollars to sequence a genome. Whereas now we're looking at potentially even like $1,000 to sequence a genome. And then coupled with that, we've really had a sequencing revolution, if you will. So the amount of data that we can now generate is, we've got billions of base pairs of data. So back in the early 80s, you might be producing 10 kilobases of data per day, whereas now, yeah, we've got lots and lots of data. And that means that we actually now have high throughput techniques that can rapidly and cost effectively capture sequence data from the environment, and we can apply this to microbial community information during mineral exploration. So Canada is a great place for us to test this idea. And that's because, as we know, many deposits are being mined out, and we're having to often find deposits that are sort of undercover. So we're trying to see through overburden, if you will. And so Canada is a great place to, you know, test some of these exploration undercover, if you will. So we've been looking at 4 different sites across Canada. So we've got 2 kimberlite sites in the Northwest Territories. We've got a Kelvin kimberlite, a DO18 kimberlite, and we've got 2 porphyries in BC that we've been looking at. We've got a copper gold porphyry and a copper molybdenum porphyry, and just big thank you to all the government and industry sponsors that have taken part in this project. And so just wanted to kind of show you a few slides on like, what does a microbial mineral exploration look like? And so it's quite similar to if you went out and did a geochemical exploration. So you would have initially sort of found the area that you want to explore. You would do some transects. You dig your holes. In this case, we always take B horizon soils across our transects because we want to be consistent across where we're looking. And essentially, you know, you'd bag a sample for bulk geochemical analysis. You know, we just need an extra 200 grams of soil for microbial analysis. So it's quite typical to what you would normally go out there to do. And then when we come back to the lab, we basically extract the DNA. This DNA then represents all the microbes that are in the soil. There's many different things, as Stephen just went over, that you can do with this DNA, but we're just focused on the barcoding part, the finger parting part. So we're just looking at one gene, and that's the 16S rRNA gene. That tells you which species of bacteria are in your sample and what's their relative abundance across the area that you're surveying. So I'm going to just show you a couple of plots from our kimberlite sites, just so you can see, like, if you were to do this kind of work, what would you actually end up as? What would the data look like? How would you define your drill target? So we're looking at the DO18 and the Kelvin deposit. So the first one, I'm going to show you what typical chemical data would look like. So this here is our DO18 deposit. The black is just showing you the outline of the deposit, and these bubble sizes and colors that you see as the concentration of the chemical element. So in this case with the chemistry, we're looking at 4 pathfinder elements: chromium, magnesium, niobium, and nickel. And as you can see from this map, they were found to be spatially associated down ice. So this is because the mechanical glacial dispersion, they're often down ice. Whereas when we do this with the microdata, you'll see that we get a much clearer spatial correlation with the surface projection of the kimberlite. And we can plot our microbial data the same way that you would look at a chemical anomaly. So these bubbles are showing you an increasing shift in the relative abundance of the microbes. And the next thing that we can do is just sum these. So I'm just showing you the comparison between the chemical fingerprint on the bottom and the microbial fingerprint on the top. And so the thing to remember though is that with the chemical fingerprint, you've only got 4 elements here. With the microbial fingerprint, this is made up of like 10s to 100s of different species, and it's only 1 gene. So you could imagine if you start to use more genes, your statistical power of defining a drill target greatly improves. And so we thought, well, we should try this at another site just to, you know, see if it works again. So this is the Kelvin site. And once again, you're looking at the kimberlite projected outline is in the black. And then you've got these bubbles, which on the chemical one shows increasing concentration of the element. With the microbial fingerprint, it's increasing relative microbial abundance. And as once again, what you're seeing is that the microbial fingerprint more clearly defines and resolves the buried kimberlite. And the thing about both these sites to remember is that with DO18, it's under 5 to 10 meters of overburden. And with this Kelvin, it's buried 150 meters down in the bedrock. And it only outcrops actually under the lake there. So we're actually seeing through overburden. And so just to kind of round off of the things that we're kind of interested in. So now that we've shown that, you know, it seems to work well, we've also done this in BC. we're actually looking at how can we make this better so that it can be used by industry. And so we're looking to develop simple and robust field sampling protocols and strategies. We're looking to build databases of background microbial communities. So that's how we get our anomalies, right? We're comparing the community over the deposit to the background. And so by having more background, a better idea of what that is, we can better define our anomalies. And so we're looking to do this in specific soil types and terrains. As I mentioned, this data is based on one gene. We're looking to expand the number of sensors we have, increase our statistical power, so develop more sensors, more functional genes. And then we're looking at integrating this with geophysical approaches. So maybe you would initially do your drone-based magnetometry and then hone in using this microbial fingerprinting technique. And so, yeah, if anyone's got questions or wants info on some of these things, you can contact myself, Sean Crow, Craig Hart, and obviously BRIM. And that's all I have.
Excellent. Thank you, Rachel. The first time I saw you present this stuff, it really hit me as something quite interesting and unique. And I get excited every time I see the presentation. So thank you very much. We have a couple of questions. Just for time, we might just hold those off to the end. They're logged and we will respond. But we might move on to the next speaker. That's Rob or Vikram? I think it's Rob.
Yep, Rob's next.
Yep.
Okay, thank you, John and Dana. And let me just get my slide set up here. Okay, there we are. Yes, so yeah, my name is Robert Greene. Thank you everyone for joining us today. We're presenting on Greene biochemistry for mineral processing reagents. Looking at more of the applied side of what is possible with some of the power of large numbers and the sequencing revolution that the previous two speakers, Steve and Rachel, discussed. So just a little overview of what we'll discuss today. First, we're just going to take a look at gold and copper recovery rates in Canada to kind of see some of the economic driving forces behind innovation in mineral processing chemistry. Take a look at the historic evolution of flotation reagents. Take a look at the emergent new era of processing chemistry. Specifically taking a look at the directed evolution of biomolecules. And more specifically, the directed evolution of mineral binding peptides. And we'll discuss mining the microbiome to identify peptide and protein-like structures assisted by AI search tools. We'll have a quick discussion about potential applications, and we'll wrap up. So starting with a look at some industry data, it's actually a pretty complete data set from the period of time 2002 to 2005, a 4-year span in the Canadian industry only. And over that period of time, the gold industry across deposit types, they were on average losing about 26% of the gold that they mined out of the ground. Over that 4-year period of time, that results in a $24.4 billion total loss. In comparison, over that same span of time, across 3 deposit types, the copper industry was losing about 14% of all copper mined out of the ground, resulting in a very similar total loss of $24.3 billion. More efficient on the copper processing side, but larger industry resulting in more overall loss. Now, at the heart of these losses is challenging mineralogy. You know, as you get out of the easiest to mine deposits and parts of deposits, and your mineralogy, mineralogical complexity increases, ore grades decrease, so on and so forth. This is an ongoing trend, though, over the history history of the life of the mining industry, and there's always revolutions and inspirations across industries that spur periods of innovation. So when you look at froth flotation processing reagents in specific, there are 4 well-defined periods of innovative evolution with an emergent period that we're just on the cusp of now. So this has all occurred over the past 120 years. And this first revolution was spurred on by developments in mechanical engineering associated with the development of the start of the machine revolution. Then you look at revolutions 2 and 3 are both due to big advances in chemical use and manufacturing, corresponding to the end of the First and Second World War. And then there's a long period of stagnation before you move into the 4th revolution that was spurred by kind of like the technology and computing revolution, allowing for more complex computer modeling and more data acquisition and analysis in regards to mineralogical composition, processing feeds, etc. Now they're in this new emergent 5th period of time. There's drives around sustainability, impact of mining, and there's also an expanse in the way that miners are being educated, as is exemplified by the cross range of various backgrounds of people presenting today, and some of the tools that are emerging out of this revolution are biochemical tools. So when you take a look at this emergent era and some of the main challenges, so as has been discussed already, there's processing low grade and poor quality ores, also maximizing resource utilization. So, getting that 25 and 14 missing percent, across deposit types, improving water usage efficiency and water resource management, which can be highly impacted by traditional chemical-based processing reagents, increasing energy efficiency, reducing waste and remediation, complying with exceedingly and increasingly restrictive health and safety and regulations, environmentally benign chemicals, and minimizing overall environmental impact in order to ensure the social license to operate. So these individuals, Dr. Nagaraj and Dr. Raymond Farinado, they are the heads of reagent development at Solvay, and what they believe are the solutions to that will dominate this new era of innovation in processing chemistry are holistic approaches, advanced statistical and analytical tools, which we see, both Dr. Hallam and Dr. Simister utilizing. And what we are proposing is the develop of biobeneficiation tools, which increase the sustainability of mineral processing with the use of greener reagents. And then we'll touch briefly on modeling chemistry and how that can accelerate the discovery and development of these tools. So having a look here at the directed evolution of biomolecules process. This is something that Dr. Scott Dunbar is one of the pioneering researchers on for the discovery of biomolecules that have a high affinity for mineral surfaces. And so essentially how this operates is you start with a library of DNA hosted in some sort of host organism, and that library is diversified. There's a number of complex methods for this. That DNA is translated into a host organism, and the biomolecule library is expressed on the outer membrane of the host organism. And then you screen this library against the target to essentially navigate through the library to find biomolecules that do what you want. So you start at a starting point and you navigate through this very complex space of molecular structures, and your path may lead you to a local maximum, that shows some capacity for performing the tasks that you're interested in, or if lucky and or patient or a combination of both, you can navigate to an absolute maximum where you have a very small handful of biomolecules distilled from a very large library with the capacity for very selective binding between different mineral surfaces. So Dr. Dunbar has previously discovered molecules for the binding of sphalerites. So as you can see here, this is the viruses that are these peptides were isolated using what's called the phage display library. So phage display are viruses that specifically infect bacteria, and you can generate and purchase very large libraries of viruses displaying these short sequences of amino acids. And by screening those libraries, Dr. Dunbar isolated particles that will, peptides that will specifically stick to sphalerite and not silica, and as well as not pyrite. So as you can see here, there's a fluorescent response built into the host cell, and while displaying the peptide, the sphalerite particles generate a red fluorescent response, but the uncoated silica particles, or in this case pyrite particles, remain dark, indicating selective binding after several stages of this phage-based evolutionary process. Sorry, moving, just realized that there's one slide that got dropped, and we'll try to maybe pull that back in like the Q&A section, showing some applications of these peptides once they've been isolated. Sorry about that. But moving beyond peptides to more complex structures, it's not a surprise that these kind of like directed evolutionary techniques with simple structures allow for the isolation of metal and mineral binding motifs, because most proteins actually are metalloproteins. An estimated of 50% of proteins actually contain metal binding motifs. Yeah, so using the biomining process and interfacing that with like the an in silico evolutionary process to speed up the rate of sifting through these lives, you can start by mining the published proteome, so databases of known protein structures for metal binding motifs. They can use in silico design tools, various software platforms, to take and modify that metal binding motif to make it integrated into a highly stable protein lattice, and also tune the specificity of metal and metal ion binding to be able to differentiate between very similar metals actually. And then from there, you can go back and produce a display library, such as on phage, of these custom metal binding proteins, for testing for mineral processing applications. Okay, and we'll wrap up there. And I will dig up that previous missing slide for the Q&A section.
Thank you very much for getting application. I think talking with Scott sometimes over coffee, I think the game changer here possibly is the critical metals, the rare earth metals, they're present in low concentration, sort of in a lot of different places and often in waste. So one things we're going to have to figure out is how to fix critical metal supply chains. Getting better with processing waste and selective processing is going to be a big part of it. So maybe that's an application of biotech again. Okay, thank you very much, Rob. We'll move on to Vikram now, I believe.
Thank you, John. So I will be sharing my screen shortly, but before I start, I'd like to congratulate you on the new directorship. This is going to be a great future for the institute moving forward. As well as I noticed Peter Bradshaw is on the line as well. So thank you to your continued support to the institution. This allows researchers such as such as the panelists today, to continue doing and having fun in the lab, and also potentially changing the world and the mining industry while we're having fun. So I'll start my screen. So hopefully you should be able to see it, and I'll go to the full screen mode. All right. So one of the big challenges with Zoom that I have noticed, especially when I've been taking lectures, is you can't really read the audience. So if this statement that I say falls flat, I do not take any of the blame. I will ascribe that entirely to Zoom. So some messes in the world, they cannot be cleaned. Okay, and the Canadians in the audience are probably chuckling when they hear that. But then there are some other messes in the world that are tailor-made for synthetic biology. And one of these things that I would like to talk about is the treatment of acid mine drainage. And I would like to convey to you how this emerging domain at the interface of engineering and biology, which we term synthetic biology, is transforming the way we design and develop processes for extraction of raw materials, natural resources, as well as extending those to the remediation of the environment. And this resonates with the mining industry, just given the span of applications that synthetic biology actually has in this industry. So before I start, and you probably have got a flavor of what BRIM and especially the subgroup on the mining microbiome is doing, is essentially applying biotechnology to address pinch points in the entire mining value chain. And this starts all the way from extraction processes. So as John had mentioned, Jetty was in the news lately, and they are working extensively on hydromet as well as biohydrometallurgy to improve extraction processes. And this is some place where biology as well as synthetic biology are having tremendous applications. And my research group actually has some work in the space of extraction as well. And then moving forward, as you start adding value to these products, there is processing. And the talk for today will address a project that we're working on in the lab that addresses aspects of processing as well as what comes thereafter, which is waste management. And of course, as Stephen and Rachel had pointed out, there are enabling technologies that are allowing synthetic biologists and engineers to design and develop applications in each of these domains. And that is, and one of those big things is, so not only are you establishing the technology platforms for each of these, but also through barcoding and other types of applications, you're extending this to environmental monitoring. But the talk for today, which is quite brief, and I'll actually give you a big picture overview of what we're doing in the domain of waste management. And you could actually be generous and extend the boxes of this particular project all the way to processing as well. And before I describe specifically what you're doing, I guess it is fitting that we describe what the nature of the challenge is that we're trying to address in the industry. So if you were to take a typical open pit mine, now there are obviously variations, but this is just based off information that if you go and calculate some of these statistics for these mines, on average, a typical open pit mine, now you could be, plus or minus these numbers, but let's assume that this mine is producing 3 million tons of ore per year. So we start right at the base of that diagram. As we realize, mining and processing are extremely energy and water intensive processes. So some of these processes and some of these mines, they end up utilizing as much as, so if for 3 million tons of ore per year, you're looking at approximately 4,000 gallons per ton of ore mined. So that translates to about 12 billion gallons of water that are being used. And such is this volume of water that has to be treated. They're essentially stored in these ponds and tailings ponds. Now, as these tailing ponds are sitting in the environment, you're having sulfides, you're having oxygen, you're having bacteria in these environments, as was described in the presentations before. This catalyzes some very interesting chemistries. and that leads to the formation of acid mine drainage. So you're having roughly about 238 million gallons per year for a typical mine. That volume of AMD or acid mine drainage is being produced. And this is extremely corrosive. It is a danger to the environment, and therefore it needs to be addressed. And the way acid mine drainage is addressed is to use quicklime to neutralize it. So you would then, for these types of volumes, you're looking at approximately 4,500 tons of quicklime. It has to be of a certain specification in order for you to use it. And that results in a total bill of about, a bill of goods of about $2 million per year. Now, the CO2 emissions that I have put here, they don't, and this is a very, very simplistic calculation that refers only to the production of lime. So every ton of mine produces about 0.8 tons of CO2. So that is just the CO2 that is being emitted from the production of lime. This does not even factor in some of the CO2 that's coming from some of the other operations, which is quite sizable. So how is it that synthetic biology is actually trying to address this? So what we're trying to do is to treat that number at the top, 238 million gallons per year, and try and come up with a better solution to that. And of course, try and eliminate some of this heavy lime utilization at these sites. So in that, as you add these numbers up, it translates to quite a sizable cost savings as well as quite a sizable reduction in CO2 emissions. Something that is also related to that is once you produce the sludge has to be disposed. The disposal cost for those volumes is roughly about an additional $500,000 per year. And that does not even factor in some of the logistical challenges that are present in that process. And then there is the other issue of regulatory concerns. So even though this is sludge, and this has been sitting as a waste stream or a byproduct stream, it is still teeming with a lot of valuable metals in there. And that represents both a concern for discharge into the environment as well as lost value. And the total concentration of all of these metals is roughly, you add them up, obviously some metals are more than others, but you're looking at about 3 grams per liter. So what is it that we have been trying to do? So in order to address this challenge, building off of the excellent fundamental as well as discovery-oriented research that collaborators such as Stephen Hallam, Sean Crow, Rachel have spoken about, Scott Dunbar as well. I have the distinct privilege of working with all of these panelists. We are essentially building off of that excellent fundamental knowledge to then try and apply it. So my view of the world is when Stephen sees a rose, he starts admiring the beauty. And when I see a rose, I essentially ask how much perfume I can extract from it. And that's just the engineering-centric view of the world. So we are using a combination of 2 processes, a microbial fuel cell in combination with another process called microbial-induced calcium precipitation. And by synergistically applying these 2 processes in that entire workflow, we are able to address all of those challenges that we are talking about. We are trying to reduce lime usage. We are trying to recycle more of the lime. We are recovering some of the lost value of the metals in those streams, which is not only a money earner, but it also helps address some of the regulatory challenges. And then, of course, you are eliminating or reducing the production of sludge. So you're saving you're saving disposal costs as well as material handling costs. So the way we're describing this is that it is both a value addition, you're saving costs as well as you are reducing expenses. So how do these 2 processes work? So research in my lab, and this is a collaboration with Scott Dunbar and his team, is we have been working quite extensively on designing and developing microbial fuel cells. So microbial fuel cells are essentially they are electrochemical cells. The big difference is that the oxidation reaction, the reaction that generates the electrons that allow for the metals to get deposited and pulled out from these wastes, that reaction is catalyzed by a special type of bacteria. They're called exoelectrogenic bacteria. So these bacteria, they consume an organic substrate, they oxidize it, produce the electrons that are then going all the way to the cathode where your metals are being deposited. Now, this process generates hydrogen and CO2. And what we figured out is you can also loop this process with some sort of a carbon fixation process. So organisms such as Clostridium are able to fix the carbon dioxide and produce the organic substrate. In this instance, it would be acetate or lactate. And then the organism in the chamber that is actually consuming the substrate is usually an organism known as Shivonella onedensis or Geobacter sulfurreducens. These are the bacteria that are then consuming the substrate and then depositing the metal. So that is the general principle of the microbial fuel cell. Our specific research is aimed at, first of all, looking at the construction of the cell, and then engineering the bacteria using some of these information, the biological, biochemical information that my colleagues are identifying, and try and genetically engineer a more productive microorganism. So among other engineering that we're looking at is we're looking at the conductivity of the surfaces of the cells, and we have done some extensive research on this field. So as we speak, we are able to actually deposit about 90% of copper in about, which is roughly about 250 milligrams per liter in about 6 days. This is obviously bench scale research, and the cells are able to generate a power density of about 10 to 20 milliwatts per meter square. So this is definitely among the higher numbers that have been published. And really what we are now doing is taking this towards larger scales. And as you are able to scale up these processes, it raises some very compelling images of how you could use microbial fuel cells to address a range of problems at the mine site. In terms of the microbial induced calcium or calcite precipitation, you're using another microorganism. So this is taking place in a second stage. You're using microorganisms such as Sporosarsina pasteuri, and that microorganism is playing off of two enzymes. So urease breaks down urea to produce ammonia and carbon dioxide. Then another enzyme, carbonic anhydrase, which is one of the fastest enzymes known in nature, that is able to then produce carbonic acid, which is then precipitating the calcium ions that are in there to produce calcium carbonate. So so how does the MICP and the MFC actually work in concert? And so before I come to this slide, so the way the process actually is it goes through an MFC cascade where you're recovering a lot of the metals that are in the stream. You use lime to neutralize that the acidity of that solution. There is a pre-treatment step where you're dissolving the gypsum in the sludge. And then once you have the calcium ions that are in there, you're using MICP to regenerate CaCO3 initially, which is then used to produce lime. And this lime is then looping back into the process. So we are trying to impose some circularity into these processes, which are inherently more sustainable and more efficient compared to your traditional linear processes. And just to show you in terms of what other extensions that we have applied these results to, we have used those same results, namely engineering bacteria to make them more conductive. This is something that we're doing to improve the performance of the fuel cells. We use that same technology to build solar cells from bacteria. So this was something that was in the news recently, and this builds off of the same foundational information that we're using in the microbial fuel cells in the MICP. So with that, as I say, the salient benefits is you're reducing waste, you're eliminating this huge tonnage of sludge, you're recovering value through the deposition of these metals, and then ultimately it translates to saved costs in terms of reducing the lime, as well as more earnings through commercialization of some of these recovered metals. So ultimately, we bring a lot of technologies to bear in order to achieve these things, and a lot of these specific domains have been discussed by some of the previous panelists and will be discussed by Susan after me as well. But ultimately, we view ourselves as the aggregators. We take all of this incredible information that is within the BRIM ecosystem, we connect it to a very clear pinch point in terms of an industrial process, and we then try and apply these things in order to develop greener processes, as is the case over here, as well as better products, more energy efficient products. This is my team. They're an incredible bunch of talented individuals. Some of them are in the audience, and none of this would be possible without their enthusiasm and their energy. And what we like to do is we like to feed off of their ideas. So with that, I will conclude. Thank you for your attention. Thank you, John, for providing me the opportunity to speak. And over to you, Susan.
Thank you very much, Vikram, and thanks for the note of congratulations at the beginning too. I'm very pleased to be in this exciting role. Now I'll hand it over to Sue Baldwin, who's I guess the leader, the director of this research theme within BRIM. I neglected to mention that BRIM has a number of research themes, of which this is one, but Sue will play a very important leadership and coordination role in this theme. So fitting that we should finish with Sue.
Okay, great. Thanks very much, John. First of all, can everyone hear me?
Yes, indeed.
Great.
You can see.
And can everyone see my slides?
Yep.
Well, if you can't see my slides,
we can.
Okay, great. Thank you. Okay, so thanks very much for joining our BRIM webinar today. I'm going to talk about how the microbiome can play a role in mine remediation, with a specific focus on how we can harness and leverage the native microorganisms that are already present on the mine site. So my name is Susan Baldwin. I'm a professor in the Chemical and Biological Engineering Department at the University of British Columbia. My interests are in bioremediation, biomonitoring, use of active and passive treatment for mine impact water, applied metagenomics and microbiology, and the circular economy, the valorization of residues for reuse and recovery of values from wastes. So these are mined landscapes. These cover very, very large areas that are many square kilometers in size. So during mining, the tops of mountains are removed, large pits are excavated, and enormous storage dams are built. These landscapes are mostly devoid of any life as far as we can see by the naked eye. So not only do we need to restore these landscapes to their natural forms, full of life again, we also must protect the surrounding ecosystems from any deleterious impacts from water runoff or any other emissions from the mine. So unforeseen events can cause catastrophic disturbances on the surrounding untouched environments, such as lakes and rivers that are adjacent to mines. Fortunately, these do not happen very often. So this is an aerial view and a land view of the physical impacts that you can see from the breaching of the Mount Poly tailings dam in 2014. And Stephen mentioned this in his talk. So even though these mined environments and the effluents that flow from them look like they don't contain any living organisms, there are always microbes present. And so Rachel conveyed that really nicely in her talk, showing the microorganisms, or the cyanobacteria, I think it was, that were utilizing iron in the oceans. So microorganisms are everywhere, and they play a very important role in biogeochemical cycling, as we've been hearing about today. They can transform minerals into different forms. They can dissolve solids. They can cause things to precipitate out. They can make acid, as Vikram explained in his talk. They can change the local environment in many different ways, and these can be good ways or bad ways. So one technique that we can use to find out what microorganisms are living in these mine environments and how similar or different they are to the pristine surrounding environments is the application of eDNA. So this stands for environmental DNA, and this is very similar to what Rachel described in her talk, is basically you walk around the mine site to the locations that you're curious about, and you pick up some soil. And similar to Rachel's pictures, you just scoop some of the soil up into a bag, a sterile bag, and then you take that back to the lab. And then you extract the DNA from that soil sample or water sample, or whatever sample you're interested in, And then you can use this DNA for all kinds of interesting things. So the DNA can be used for biomarkers. So in Rachel's talk, she was looking for biomarkers of the presence of minerals. Here, when we're addressing a catastrophic disturbance, we're looking for biomarkers of the disturbance. So in this plot, here's an aerial view of the Mount Polly area, some natural lakes and the area, disturbed areas, and you can see that one particular organism, this Thiobacillus, was growing quite happily in the disturbed areas. It's a natural microorganism, it's found everywhere, but it seems to very much like the environment of the deposited tailings. And so this is important because we can begin to understand what underlying biochemical processes are taking place. And knowing what types of organisms are there and getting the metagenomics piece that Stephen talked about, we can identify the good and the bad actors. And then most importantly, we can identify the microorganisms whose metabolic potential can be leveraged so as to remediate the area, the mined and the disturbed sites, and allow for ecological restoration. So this is just an example of bacteria that could be considered bad and good in the context of a mine environment. They're not always, I'm not saying they're always good or always bad. In other contexts, there are many places where both of these groups are very important. So in a mine environment, sulfur oxidizing bacteria can cause sulfide minerals, which are solids, to dissolve. And in so doing, release potentially toxic elements such as arsenic and create a very hostile environment, such as acid. On the other hand, in many mine environments, sulfate-reducing bacteria are considered good bacteria. They take sulfate and reduce it into sulfide, which reacts with these metals in solution, causing them to precipitate out as sparingly soluble metal sulfides. So the objective in mine remediation is to engineer our systems in such a way as they favor the good bacteria over the bad. So it's always best to start mine remediation at the source. So for instance, coal mining waste can leach out an element called selenium that can be very damaging to aquatic life. So other than just stopping mining altogether, it is best to prevent the release of selenium from the waste rock piles. That involves learning about what types of bacteria already exist in the coal mine wastes. Are they good or are they bad? Where do the good bacteria live? And can we transfer them to replace the bad bacteria that are accelerating the release of selenium? By engineering the waste rock environment, we can take action to prevent the bad bacteria. For instance, limiting the oxygen that they might need to breathe. Or we can create an environment that favors the beneficial microorganisms, doing things such as supplementing the good bacteria, bioaugmentation with the bacteria with the metabolic potential we want, encouraging the formation of biofilms, adding special nutrients and other things that can alter the conditions inside the mine waste. All of this requires knowing what the microbial community is inside the waste and how to work with it in order to shift the community towards more favorable functional traits that prevent metal leaching. So we still only know less than 1% of all the microorganisms on Earth. In order to leverage the metabolic potential of microorganisms that can aid in mine remediation, we're prospecting for microorganisms at mine sites to select for consortia that have the capacity to perform the functions that we desire. For example, such as bacteria that can change selenium from soluble forms to insoluble forms and pull the selenium out of the mine-influenced water. So to know exactly who is capable of doing what, we use the Metagenomics tools that Stephen told you about at the very beginning of this talk. That allows us to take the DNA from our sample, and then just from that DNA, we can assemble genomes for each of the species that are in that sample. And then we have a good idea of what microorganisms are there, and not only that, what are the functions that they can perform. And that might be functions important for mine remediation, but it also be functions that are important to know how to cultivate them. And I should also note that we're often looking at consortia of microorganisms. It's not one individual species that that can achieve all of these goals. It's a group of microorganisms working together in a community. So despite our best efforts to prevent metal leaching, mine-influenced water can be formed on mine sites, like the acid rock drainage that Vikram mentioned. And again, we have the opportunity here to leverage the metabolic potential of the mine microbiome in order to engineer treatment systems for removing the toxic components and rendering the water safe for release. There are many different types of engineered systems that can be used, from so-called active processes that use tanks, pumps, process control, to so-called passive treatment systems that leverage natural biogeochemical processes. In many cases, the required microbial communities with the desired traits are already in the water to be treated, and within the engineered environment, conditions are created to favor those that contribute to treatment. Sometimes these systems don't work properly, and then we can use metagenomics as a diagnostic tool to understand what the metabolic potential looks like when they're working properly, and what it looks like when they're not working properly, and do that comparison that Stephen showed, and then we can tease out the actual causes for that disturbance and do something about it. So what is the future for mine remediation? So we're making use of opportunities to interact with other industries. And here we can use residues from other industries on the mine site to do mine remediation. So this diverts these residues from being landfilled, which is what is happening currently, where they pose a liability to the companies. Also, we are developing new hybrid active-passive mine-influenced water treatment systems, specifically so that we can recover values from the mine water, such as metals and also nutrients. This not only removes these liabilities from the mine site, and it also enables their reuse in other applications. So overall, mine water treatment can produce clean water for reuse and therefore reduce the dependency on the ever-diminishing freshwater supply. So these new hybrid active and passive treatment systems can be created with the elements of process control to properly leverage the metabolic potential of mine microbiome or the metabolic potential of the synthetic microorganisms that Vikram is going to create. So that's all for me. Thanks very much for listening, and I'm really delighted to answer any questions that you may have.
Thanks very much, Sue. Great presentation, and also the vision for mining being part of a circular economy. It's really the holy grail for our industry. So we've got some time for questions. Perhaps Dana, if I could ask you to moderate the questions in the list, and we'll just work through that. And if you've got any other questions, we do have 15 minutes before we conclude at 1/2 the hour.
Yeah, no problem. So I'm going to go back time-wise a bit. So some of these are back to Rachel's talk. Can you apply exploration of microbes to space, e.g. Mars?
Never really thought about it. I guess we're using like live microbes, and never really thought how you'd use that on Mars, unless you're talking about like ancient signatures from microbes if they were on Mars a really long time ago. I don't know. Anyone else have an idea?
I'm thinking of the David Bowie song "Life on Mars," but that's about all I can think of at the moment. It's interesting to see what might come out of that exploration. But yeah,
there was just a paper that came out in Nature Communications about cores that were tens of millions of years old, and they were able to get microbes to grow from those. So maybe on Mars.
Yeah,
cool.
Yeah, good.
It's an interesting direction, yeah.
I have a perspective on that. So, I mean, microorganisms are incredibly powerful. The versatility of some of these reactions is immense. So the general answer is, could you apply microorganisms to exploring resources on Mars? The answer is yes. But the big challenge is, are we in a position to actually collect that information and actually interpret it correctly? And it's that lag in communication that's something that needs to be developed. there is some information. There is some very exciting research taking place. People are doing experiments using microorganisms on the International Space Station, for example. But generally, it's the way we work is it's a very physical process in that you collect the microorganisms. There is that the physicality of actually putting it in an environment to study it, and being able to do that remotely using some very interesting robotics, and to communicate with the robotics is going to be the bottleneck step. But I believe there are companies, including NASA, that are already doing this. So it's not too far fetched.
Interesting.
Interesting.
We might move on to another question.
Yeah, so the next one I have is also for Rachel. Thanks so much. Were the indicator species the same for the porphyrin kimberlite? Do you quantify qPCR, the 16S gene? What other genes are you expecting to use?
So right now, because of the different geology, we've actually kept porphyries and kimberlite systems separate. But within, say, the two kimberlites, there are indicators that are shared between those two kimberlites. And then in our porphyries, there are indicators that are shared at those different field sites. When we eventually, I have thought about combining everything together. It's just for now, we were treating them as these are quite different geologies, different deposits. We'll keep them separate. But it's something I'm going to look at out of interest. And then quantification, yeah, we definitely want to do that. We have samples stored. We just haven't got around to it yet. And there was another part that I don't remember. Oh, other genes. Yeah, so we were thinking about, I guess, more at, some of these organisms have things like copper monooxygenases in them, or like looking at sulfur cycling. If they're doing sulfur oxidation in the deposits, we could look at, there's certain known genes that do those processes. So the idea would be, if that's what they're kind of doing, maybe that signature would be there. But one of the things we still have to answer is, what is the mechanism? Because I talked about how these signals we're getting are at the surface, but the deposit can sometimes be buried like 10 to 50 meters down there. So we need to also figure out, what is the signal we're picking up? Is it response to gas that's being given off? And then that might be a different set of genes we'd look at. So yeah.
All right, sounds good. Thanks.
Excellent.
I'm not sure who this one is aimed towards, but could you please explain in a bit more detail what SUM, capital SUM, means in the case of microbial fingerprint?
Oh, that's aimed at me. Some of the maps that I showed, so when I show the geochemical analysis, we show the elements separately, and the same for the microbes. I was giving 4 individual species, but then we actually just basically amalgamate that data together to have one big plot. So the SUM map for the geochemistry is a combination of the chromium, nickel, niobium, manganese data, and then for the microsum, it's a combination of, say, 10, 20, 50 species all grouped. So you just get one plot, instead of, with the microbes, you could imagine if you had 1,000 microbes that indicated on the deposit, you'd need to look at 1,000 maps. So we just sum them all together, basically. So you get one.
Sounds good, thank you.
Okay.
The next one, I think this would probably be directed towards Rob based on the time. Do these biomes cause any corrosion problems? Or if someone else has an answer to that as well.
Yeah, so
guess,
in regards to looking at mineral binding peptides, as small amino acid chains, yeah, they're essentially biodegradable, so there wouldn't be any resultant corrosion. And in fact, we've actually looked at them as something to remove corrosion causing materials. So it's kind of more of the opposite, actually.
Sounds good. Since we're on Rob right now, Scott Dunbar had the slide that you had dropped, and he wanted to talk about that for a second as well.
Oh, thanks, Scott.
If you should be able to share your screen and put that up. Scott, were you wanting to go through that? Oh, there we go. I think you're muted.
they're tagged to the chalcopyrite particles, the dark particles here, and the clear particles here are silica. Although there's a clear selectivity for chalcopyrite. I'll let Rob describe this. Next slide. Rob?
Yeah, so Scott isolated these chalcopyrite binding peptides with selective binding between for chalcopyrite and not for silicate materials from a phage display library. And then we needed some way to actually use them as a separation mechanism. So what we did is we attached the peptide using a covalent cross-linking polymer to the surface of iron oxide nanoparticles, which are strongly ferromagnetic, and they would selectively deposit on the surface of chalcopyrite, and they could be recovered with a magnetic field. In this case, just a neodymium rare earth magnet taped physically to the side of a test tube, and at a ratio of about 20 times mineral to peptide, we had very efficient recovery of sub 40 micron particles. And these are particles that are often missed by traditional processing methods. That's why they're in kind of that 14% of copper that does not get recovered. But it also happens to be acid generating. So there's options to kind of go for a double kind of like waste to value and remediation play with this sort of approach. Thanks, Scott.
Actually, I have another question for you. Do these bugs affect downstream processes like CIP or CIL?
So that's actually been a question that we've been asked in the past. And the answer should be no, where if you had the more caustic solutions further downstream, they should essentially just kind of break apart the small amino acid chains, which are meant to be primarily operated between a pH of kind of like 5 and 9 as our sort of maximum range. So downstream processes should remain unaffected.
It's important to point out, I think many of you know it, that these peptides are not live organisms. They're just strings of chemicals, large chemicals, they're large pieces of macromolecules.
Right.
Strings of amino acids, Scott, maybe.
That's what they asked. That's it.
I knew that biochemistry degree would be useful at some point, some decade. Yeah. And we have another, I think one more question.
Yeah, I think Vikram responded in the comment, but I'll just read it out,
okay.
And then we'll wrap up after this one. Is it possible to increase the speed on how bacteria extract metals from ore in bioleaching? So,
I can take that. So it depends on so if it's so I guess this question is how selective bacteria to extract. So in the case of bioleaching, where you're using acid catalyzed processes, the selectivity is low because the acid it affects the other things equally. So then it depends on the thermodynamics of the reaction for every individual metal that's being leached out. In the case of electrochemical systems, it is also the same, but over there you're looking at half cell potentials when it comes to depositing or dissolving out these metals. So there are some, in the case of bioleaching, it is harder to control. In the case of electrochemical processes, there are some very interesting electrode synthesis techniques. In fact, some of these challenges have been addressed in the fuel cells industry, and those have been used to improve the selectivity of the extraction, of the leaching, as well as the deposition.
Thanks, Vikram. I think we have one more question there about the selectivity of specific metals from ores.
Oh, that was the one.
Okay, that's yep.
all right.
Getting confused with the peptides. Good. I think that concludes. So I'll just wrap up by saying thank you to all the speakers. It's really great to get us out in public in one space, because it just tells a really coherent story, and there's narrative there about how this all fits together. And even though it's individual labs, but you know, there's communication. We solve big challenges together. So that's great to see. To the audience, as I said before, we're open to discussions about potential collaborations. As this was going on, there's actually a few emails which have come in, which is fantastic. But we're definitely open to discussions. That getting out into industry is a mission of BRIM. And also, we're revamping the website of BRIM. It's being updated, and there'll be a new microbiome page, and all of the presentations, the segments will be posted there, including all the information about the microbiome group and the microbiome's work. But thank you once again. Thank you for attending. I know things are busy. We'll continue to post more information as time goes on, and there'll be more launches of research themes as 2020 progresses. But exciting times for the mining industry. I saw today that even though things are pretty tough with COVID and the logistics around that, EY's Canadian Mining Survey shows that mining activity levels are now better than, more active than they were pre-COVID. So think it's really up to the mining industry, I think, to lead the general economy back into recovery. So we really hope that innovation and research and development can be part of that story. Great, thank you very much. Enjoy the rest of your day.
Thank you.
Thank you, everybody.
Thanks, everybody.
Thank you.